part of what I'm trying to do is trying to introduce you to some of the, basically the biology, so that when you hear the talk tonight or this afternoon, you have some idea, at least things will sound familiar, and you'll kind of have an idea of what's going on. So it's going to be a little bit of very brief introduction to molecular biology, a little bit an introduction to the androgen receptor, a little bit of some molecular genetics, and then a little bit of kind of showing you how great the KDA has been on researching grants and how successful we've been. Yes, this is a picture from a long time ago, and these are me and my two cousins. All three of us all have KD. About 11 years ago, somewhere in 1999, I got a call from my sister telling me that one of my cousins had this disease that I never heard of, called Kennedy's disease. It was something that was inherited through his mother, and his mother was my mother's sister. And I immediately went to the web, looked up, and what I found was Terry and Suzanne's site. I'm sure most of us at that time, that was the main place we got information. I read the site. I knew right away I had it. I didn't think I had it until but all the little things I knew, so that was kind of my nightmare on, well, we live on Peregrine Falcon, so that's Now, essentially, KD now is essentially a mystery. And so, back in the old days, you used to read Hardy Boys, and I don't remember this one, but I saw it on the internet, so it must have been there. And it's a mystery of trying to find out what's happening, what's going wrong. So you always ask questions and mysteries. So we're going to ask, what's happening? Well, we all know that our muscles are basically atrophying. They're getting very small, very weak. We have trouble walking around, and you know all that sort of stuff. The reason that's happening used to be clear. Now we're not as clear as about it. It's one of the sad things about research, that the more you know, kind of the more difficult to interpret. What we do know is that muscle cells, which are these little red things, down in that side, are going to be falling apart. They are getting very small, very weak. They receive input from these cells here. These are nerve cells called motor neurons. And it had been, for example, here is a motor neuron, and here are the little terminals on the muscle cells. So the muscle cells are the kind of shadowy things right in here. And then you can see they all have a little terminal, and that's how they communicate. When you want to move your muscle, you say you say move. Your brain decides which neurons to activate, and then they cause your muscle to contract. It takes about, a couple of microseconds. So that's pretty cool. Now, what's happening is that at least what we thought was happening, was that the motor neurons in the region that's the orange, that's this pons, medulla, and spinal cord, don't need to know the names. You can kind of almost imagine, you got to use your imagination, and this is a bad pun, but if you look at this, it almost looks like this is a swelling up here. It kind of looks like a light bulb, and then it comes down to where you screw it in. So this region up here is known as the bulbar region. And this is the spinal cord, and thus we have spinal bulbar muscular atrophy, because our muscles are falling apart, and it's the muscles that are controlled by these regions. And basically, that's all the skeletal muscles. So the bulbar region controls the muscles of the face and the neck. The spinal cord does basically all the rest. And we're losing muscles from each of them. Now, this is just another picture of the same sort of idea. That here's the motor neuron. The motor neurons lie, let's look in the spinal cord, right in there, and they send little axons out at these processes. They send them to the muscles. This is what a spinal cord looks like from the cross-section. So now you're getting some anatomy, so it's really exciting. And when I took anatomy, the first thing I was told when we looked at the spinal cord is that you had two different parts. You had this outer region, which although it's orange, is known as the white matter, and the inner green matter is called the gray matter. The gray matter is the part that I'm interested in, for you to be interested in, I should say. And the gray matter looks kind of like a butterfly, sometimes. You have to use your imagination. And there are special names to different sections. And the section I'm interested in is right in here, and that's called the anterior horn. The reason I want you to look at it is because this is a picture of a real spinal cord. This is that butterfly outline. You can kind of see right here. This is the gray matter. The white matter is all this stuff. And if you look at that anterior horn, you see lots of little purple spots. Actually, they're very large purple spots. And if you blow one up, this is what it is, and that is the motor neuron. The motor neurons are one of the largest cells in the spinal cord. This is taken from a person who does not have KDA. If you have someone who does have it, you can see, now this is only one half of the butterfly. This is because it's the right half, so this is the anterior horn. And you notice there aren't very many of those purple dots anymore. In fact, it's very rare you're ever going to see a scientific paper that has arrows pointing to nothing. Usually they're pointing to something. And so what was thought to be happening was that the motor neurons were dying. Now when motor neurons die, what happens is this is supposed to represent by, this is called falling apart, the muscle cells atrophy. This is known. I mean, they've done experiments in rats and tried to do things where they would actually, cut them, and they could see the degeneration and the atrophy of the muscle. So it was thought that that was happening, but in the last couple of years, there's been a couple of studies that kind of suggest, well, that may not be quite true. And so the bottom line is, it might be that the muscles are degenerating first, and that's causing the motor neurons to degenerate, or it might be the reverse. So we're not. It's terrible. 2 years ago when I said it, we were very certain, and now we're not certain. So, nonetheless, we still know we're losing motor neurons, we're losing muscles, we just don't know, is it the egg or the chicken? So, next question is how? Because it's a genetic disease, and I think with Cliff's talk you had a nice idea of what the genetics were. When we think of genetics, people who aren't in biology, they think, well, we know that's inherited, and that's true, and that's why people care about it. However, when a scientist hears about it, that is a clue to what's wrong with the disease, what's causing the disease. If the disease, for example, was caused by a bacterium, we know we can kill it with antibiotics and cure it. But being a genetic disease, this is something we're born with. And we know that what's wrong is that we have a protein that's defective. Okay, and we'll try to explain to you why we now know that. When you think of protein, of course, most of us think of this, unless you're a vegetarian, then you think of that. That's not the way you have to think about. It's not the way to think about proteins in biology. I use in my class sometimes, especially in introductory class, an analogy that a body is kind of like a city. And each of the buildings are like little cells, or each of the cells are like buildings. And each building is different, not necessarily, some of the same, but many of them different, and they have different functions. What makes, let's say, a car factory different than, let's say, a distillery is that they have different machines on the inside. The outside could look very similar, but the inside has different machines. The machines themselves are the proteins. And so you shouldn't think of them as food. It's not today. You should think of them as appliances. So these are kitchen appliances. So there might be a bunch of proteins in one area that are kitchen appliances. Or there might be tools. So we went for them male, female. But, televisions, whatever is inside your house that does something, in a cell, that would be made by a protein. You can make something on the order of around 50 to 70,000 suspect different proteins. And just like a fine-tuned machine, if you make the machine poorly, the machine doesn't work right. If you don't make the protein right, it won't work right. And what's even worse is that proteins, I don't know if you ever watched, it's Alton Brown on the Food Network. And he always talks about tools in the kitchen that only have one function. He calls them, he has see now I forget the name. He has a special name for a uni machine or something like that. But they only make one, and he thinks that's a waste. Well, proteins are exactly like that. They only can do one thing. So if you're missing that protein, then you're missing some function that's supposed to happen. Sometimes it doesn't make any difference. I have blue eyes. That's because I'm missing a protein that makes a brown pigment. So the fact that I don't have that pigment only means that I'm lucky that I have blue eyes. But other times it can be much more serious. Now, why being a genetic disease do you know that that's necessarily going to affect the protein? So we need to talk a little bit about some molecular biology. These are just some pictures of what some different proteins look like, just to show you that they're not just simple machines. They're very, very complex, some of them. And if some of you who were with me the last time when we talked about the proteasome, which is the thing that was the wood chipper, this is what it, whoa, what that was the wrong one. That's the proteasome down in there. But then there's all these other proteins. That's it. Every protein looks different. Every protein is built different. Now, if you want to build a machine, you need instructions. If you want to build a protein, you also need instructions. Proteins are built out of relatively simple molecules, 20 different of them. They're called amino acids. And these are the 20 different amino acids. The only reason I only showed you this is because eventually today someone's going to mention an amino acid by a single letter. And this has the single letter abbreviations. Or arginine is an R, and you molecular biologists get very lazy when you're writing. They don't want to write out the whole, and you'll understand why in a couple of minutes. So these single letter abbreviations. What makes each protein unique is the sequence of amino acids. It's almost like spelling a word. If you want to spell a word, you have 26 different choices of letters. But if you want to spell a specific word, you have to use the right letters in the right order. A long word would have maybe 12 letters in it, 15 letters. A small protein would have 100 amino acids. And you would have to have it in the correct order with all 100 of them. So imagine, where do you go to spell a word? And you go to a dictionary. Where does the cell figure out how to make a protein? And this, never mind this. They're going to go to your genes. Your genes are the instructions on how to make proteins. So therefore, if we have a genetic disease, we must be making a protein that is defective. And up until 1991, we had no idea what that was in Kennedy's disease. And in 1991, Dr. Alice Potter, who's going to be here, hopefully, today, in Kurt Fishbeck's lab, who also hopefully is going to be here today, I'm assuming, were able to discover that the protein that was defective was a protein called the androgen receptor. So now we know what's wrong. We don't know why it causes diseases, but we know that the problem is that the instructions that we have for making the androgen receptor are defective. We have a mutation in our genes that causes us to make an androgen receptor that isn't quite normal. Now here's a picture of the androgen receptor, but we're gonna have a better one. Now, it's hard to imagine that this is what it really looks like in certain instances, and the one we have is going to be a cartoon, but this doesn't help us understand anything about what it does, because it doesn't help me, because I don't know enough. But it's a pretty complicated structure. It has, like I said, a series of amino acids in there, and these are the amino acids. And so remember we had single letter abbreviations, so the first one, you can go through, and if you knew the letters, you can now list all the amino acids. There's a little bit over 900 of them, I believe. So it's a pretty good sized protein. And you can imagine that if you want to make it correctly, you've got to spell it right. So think of a spelling bee where you have to do 900 letters in a row and get it right every time. So, that's pretty good. And who does that is, that's what your genes do. That's what DNA does. Now, we have to do a little bit of, how do you make a protein? Now, if you think about it, to make a protein, you need two things. You need the amino acids. That's why you eat proteins, so you can get amino acids to make your own. So that's why we have the meat and the tofu. And we need the instructions. That's the bare minimum. Well, the instructions, as we've just discovered, are in the DNA, in our genes. That's hidden inside a structure called the nucleus. Most of us have heard of the nucleus on the side. But the structure that actually makes the protein is actually not in the nucleus. It's a thing called a ribosome, and it's in the cytoplasm, the part that's not nucleus. The bad news is DNA is too big to leave the nucleus. The ribosome is too big to go into the nucleus. So now we have a problem. How do we get the two to meet to make a protein? And the answer is, we copy the DNA. We don't copy the whole DNA. We just copy a small segment that has the instructions that we're looking for. Just like when you're looking up how to spell a word, you don't look up every word. You look for the particular word you're interested in. So if you want to make the androgen receptor, there are mechanisms by which the cell can find the instructions in the DNA. They make a copy of it in a form of a molecule that you don't have to know, but I'll tell you anyhow, called RNA. That process is called transcription. Because you may hear that today. The RNA is now a small piece of base. It's like a Xerox copy of the gene that we're interested in. It's able to leave the nucleus, go to the ribosome, and then the ribosome somehow takes what's on the DNA, or excuse me, the RNA, and makes a protein from it. And they're able to make all that copying perfect. Well, hopefully. So that you make the protein perfect. And it works pretty well. So, if we were to have a test today, you would be expected to remember that to make a protein, you have to find the gene in the DNA, you have to make an RNA, there's no way you know this, you have to memorize it, in a process called transcription, because you're transcribing, you're copying. And then you're going to use the RNA to make the protein, in a process called translation, because you're going from the world of nucleic acids and RNA to the world of proteins. So somebody thought that was a cute metaphor, saying translation. Does that make sense? Okay, so one of the questions, and you'll understand why we're doing this, one of the questions you might have but don't want to tell me is, well, how does DNA tell you what the sequence of amino acids is? And probably you don't really care, and you're probably hoping maybe he won't tell us and then we can go on to something else, but we're going to move on. DNA, like proteins, is a long chain of repeating molecules. When there are 20 amino acids, there are only 4 molecules that make DNA. They are called nucleotides, and there's a U nucleotide, a G, a C, and an A. Now, I have to tell you, if you've had biology, you know that U is only an RNA, so I'm really showing you the copy. In DNA, there's a T. It's the same thing. I probably should not have said that. Now, in looking at it, it's like a code. It's like, during World War II, trying to break a code and trying to say, well, how does this tell us what amino acid is which? So in the 1950s, 1960s, after about 12 years of work, basically throughout the world, they were able to solve the mystery. And the basis of the mystery is that in nucleotides, the sequence is divided into 3-letter words. And so every 3 letters represents an amino acid. So the first thing you do if you had a sequence is break it off into 3-letter words. So UUU, GUC, AAA, UGA. Now realize this is very short. A real gene would have, could have thousands of these nucleotides, and you'd have to break them off into 3. Then you have to say, well, what does UUU stand for? And now you need the secret decoder ring. And I just happen to have a copy. And notice, if you find UUU, it's next to the amino acid that stands for phenylalanine. So that means the ribosome knows wherever UUU exists when it's copying that piece of RNA. It says that's where phenylalanine goes. Whenever you have GUC, it's something called valine. Anytime you see AAA, that was the 3rd one, it's lysine. And you can kind of see, if you just look around, that all 20 amino acids are located here. And all of them have a code. Sometimes they have more than 1 code. So it's, right? Could be very confusing. And then UGA says stop. You say that's an interesting name for an amino acid. That just means you're at the end of the protein. And that's fun. You stop making it. So if we go back to our sequence, our UUU, GUC, AAA, UGA, we now know by using the code that that's coding for phenylalanine, valine, lysine, stop. And if you have the code, anybody can read it. And what's both amazing and really cool is that, except for very few exceptions, every organism on Earth uses the exact same cheat sheet. So a gene that's in a bacteria can be read by human cells. Or a human gene can be read by yeast cells. It can be read by, it's all in the same code. There's other problems about doing it, but the code isn't the problem. And that's going to allow us to do really cool things. Well, we don't do it, but people in the back do. That I'll talk about later. Well, remember we had the long list of amino acids? This is the list of the nucleotides in the engine receptor gene. Since there's about 900 amino acids, and you need 3, there's at least 3 times. There's around a little over 2,700 nucleotides. And there it is. So now it's a little bit tougher to spell because there's a lot more. Now, What we do know is that if you want to alter that sequence, you're going to alter which amino acid you put in. It's because you're going to change the way it reads. The cell doesn't know. The cell can only use what's there and can only make based upon the sheet. It can't think, oh, he didn't mean to put that. It can only do what's there. And so I have a little thing that I do for my students to kind of show the different types of changes that DNA can have. But since using nucleotides is kind of boring, we have a sentence. But we use the same idea that we have to have 3-letter words. So if I ask you about 3-letter words, you'd be able to say, hey, the cat ate the rat. End. Well, end is like a period. So that's pretty easy to read. One sort of mutation that can happen in DNA is you can have a substitution. You can take one of the letters and put something else. In DNA, it might be changing the A to a G. Now, and here, let's say we did put the C in cat to K in cat. Now, if you read that, if you didn't see the first one, and I said, what does that say? You'd say, well, it says the cat ate the rat, but you're not a very good speller. And there probably isn't anyone who wouldn't be able to recognize that I meant to say cat. So sometimes when you make a mutation, it doesn't really change the protein. The meaning is still there. So this would be a relatively harmless mutation, because we couldn't tell the difference. We still know that who ate the rat. And this one's a little bit tricky. Kind of depends on your education. Maybe, say, 50% of the people might realize that, well, Q in Romance languages and Latin kind of has a K sound. So they say, well, maybe what they meant to say was the cat ate the rat, but boy, they really screwed up big time. So this would be a protein that when you made, it changed it, but it still worked, just didn't work as well. So that could happen in substitution. And then you have the worst case with regard to substitutions, and this is where you change the meaning completely. Now there's no cat eating it, it's a bag. So the meaning of the sentence is very different. There's no way you'd be able to get back and realize they really meant cat with that sentence. So some of those mutations are much more harmful than others. Two other types of mutations are when you have a deletion or an insertion. In other words, you cut out. Here I cut out the C, and here I added an extra base that got put in there by accident. And that can happen. Look how this happens. Now try to read this one with the rule. You have to do every 3 letters. So it's the, ata, tet, her, at, and there's no meaning whatsoever. And the same is true with the other one. It's the, sha, tat, eth, ra. No meaning whatsoever. So when you have that sort of mutation, the protein you get is completely useless. It's not even close to being right. It does nothing, and it will actually be immediately probably destroyed by the cell. This is what happens for people with people with blue eyes. They make a protein that doesn't work at all. It's that thing does. It's going to work for them. You might be wondering, what about people with KD? What sort of mutation do they have? And hopefully one of your big questions might come out. They don't have that at all. They have something completely different. They have a stutter. They do the cat interact. So you still have the meaning, but you got a lot of cats there. This is called a repeat, because you repeat the word over and over and over and over and over. Now, I spent almost an hour at work. I don't think it was worth it. But it's still a horror movie. It's kind of good. What did I do? There we go. This is the sequence of nucleotides. If there's a repeat, we should be able to see that repeat. Like we had cat. And there it is. Right there. Now, it's hard to see back there. That's blown up. And what it is a repeat of the same 3 letters. C-A-G, 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 C-A-G. This time, 20 times. This is known as, probably you now guess, the CAG repeat. It's the number 1 question someone has when they find someone, what's your repeat? And the interesting answer is the number of repeats for everybody varies. My wife has actually 2 copies. She has 2 different repeat rates. I don't know what they are. She could have 16. She could have 25. The repeat length in everybody's gene varies. What Dr. Laspada discovered was that anybody who had a repeat length of greater than 40 had Kennedy disease. Anybody below, I think they said 35, didn't. I guess there's a little gray area in the middle. So what was causing the disease was apparently a very strange genetic mutation. It had never, as far as I know, been seen before. This sort of trinucleotide repeat, or CAG repeat. And so, I mean, my number is 46. That's above 40, so that's bad. But that's what your CAGs are. And of course, remember how you say it, CAG. Okay. Now, you might wonder, gee, if that codes for an amino acid, and CAG is one of the code words, what amino acid does it stand for? Well, it stands for the amino acid, glutamine. So if you go to the protein and look at the sequence of amino acids, you should find an area where there are 20, if we had 20 CAGs, 20 glutamines. The abbreviation for glutamine is, of course, logically a Q. I have no idea why. I think G was already used, and after that, I'm not sure. But anyhow, if you look there, there are 20 Qs. So, in my protein, I have 46 Qs in that region. Now, in Latin, poly means many, so this is sometimes also referred to as a poly-Q disease, and this is why it's a poly-Q. And then there's a lot of puns with that. Like there's one of the punning agencies is high Q. Now, what's even more interesting, well, you may not think so, is that we're not the only disease that has a poly-Q. There are at least 8 other diseases that have poly-Qs. Most of them, like Cliff was saying, you've never heard of. There's all these spinocerebellar ataxias. They can't even afford their own names, you know, it's all the same names. But there we are, with Kennedy's disease. But there is one famous one, and that's Huntington's disease. Okay, and all of them are a poly-Q disease. They all have that increased CAG repeat in the middle of their gene, which causes them to have a poly-Q region inside the protein. But each one of those is a different gene, so they affect a different protein. So what makes Huntington different than, let's say, SBMA or Kennedy's disease is they affect a protein called Huntington. And we don't know what Huntington does, but we know that when we have a CAG repeat in their cells die. We don't know why. In Kennedy's disease, we know it's the androgen receptor. So that's kind of good and kind of bad, that all these diseases. It's good because since they all affect nerve cells, and they all have that expanded poly-Q region, there must be something in the mechanism that is common between them. But on the other hand, Huntington's disease is not SBMA. They affect different cells. So there must be something specific about the protein involved that also affects the death. And why in one cell and not another cell? We're actually unlucky ones. We have one of the milder versions of the CAG repeats. So we're going to see that if people try to study it, we have 2 major problems. That we have to explain how all the CAG repeats cause death, but why they only cause death in certain cells and not others. And we don't know that answer. What do they think it's doing? Good question. They don't know. So this is a slide I stole from someone. And what it basically says, if you have a poly-Q expansion, that's the fancy term for poly-Q, you're going to get a protein that's going to fold a little bit differently than the normal fold. It's kind of like having a stretch limousine. You know, it looks like a car, but you can tell that it's not a normal car. And that may have a number of different types of effects. It might be that it affects the way the protein works. So maybe the problem is by putting in the poly-Q, the protein doesn't work right, the cells die because they're missing that activity. That does not seem to be the case in any of the poly-Q disease directly. Doesn't mean it doesn't play a role, but it does not seem to be. It could be that proteins, although they're unitaskers, that's the word I was thinking of, they also are team players. They play with other proteins, and they play very well, and very specifically. Maybe by changing the structure, they don't play as well. And somehow that leads to degeneration. Or maybe they just somehow cause other proteins to misfold. I mean, we just don't know. It could be a mixture of those. So, the bottom line is we're not going to know at this point why they degenerate. That's the people in the back are going to tell us this afternoon. But those are possibilities. Everybody okay so far? for Kennedy's disease, it affects the androgen receptor. This is a short list of all the things that testosterone is the main androgen, androgens are the male hormones, testosterone and dihydrotestosterone. So if the androgen receptor did not work because of this mutation, then males would not have any response to testosterone. And they would have none of these. There are people who are males who don't make a working androgen receptor. And they have none of these. In fact, if you saw them, you'd think they were female. I'm hoping that for all of us, most of you realize that's not us. On top of that, we have proof. This is my daughter, one of my daughters. This is my other daughter. If you met them, there's no doubt they're mine. So I know that Kennedy's disease, even though it affects the androgen receptor, does not make the androgen receptor not work. So what that means is there must be, when we get that CAG repeat, there must be something kind of added to its function. And that added function kills the cells. And so you'll hear what's called a toxic gain of function mutation. Count how many times that's said this afternoon. What that means is that the protein that is normally good still works, but in addition to that, it causes something else, which we don't know, that kills the cell. And so that's what's happening to us, is the androgen receptor is doing something different and something extra to kill the cells.
Couldn't it be something less?
No, because they work just fine. In other words, we're still male, we still have kids. So all the receptors still work. Now, as we get older, they don't work as well, and there's probably reasons for that. So it may not work as well. It might be like putting a Q in that sentence. But it still works good enough to have kids. Now, that doesn't mean that the function of the protein is not involved. It just means that we're not exposed to a lack of function. Every year I try to bring up something new that I think the scientists are going to talk about. And this year, I think they're going to talk about some of the biology and the biochemistry of the androgen receptor itself. And remember we saw an androgen receptor before, it had all those curlicues and built around, and this is a much better androgen receptor. And you're going to say, it's a Swiss Army knife. Well, it looks like that, and it's pretty harmless then, but what can a Swiss Army knife do? It can open up and you have all these cool tools. The androgen receptor seems to act just like that. And each of the things that unfold from the androgen receptor, you're going to learn that, you won't be quizzed on that, have their own special function, just like you have a bottle opener and scissors and, is that a saw? So here's my very bad picture of an androgen receptor. So let me talk you through it. And there's going to be some biology, microbiology, and you're going to have to get used to some of these letters. Because microbiologists love abbreviations. 3-letter abbreviations, so that when they speak, there's no verbs, there's no nouns, just GBD, ATT, AKT, MEP31 did it. And we don't know what they mean. So this is supposed to be a very bad picture of a cell. So here's the nucleus, we know that's where the DNA is. Here's the cytoplasm, that's where everything else is a cell. Here's outside the cell. Here is the androgen receptor. The androgen receptor, when it's not doing anything, in other words, there's no androgens around, no testosterone, no dihydrotestosterone, is like the closed up Swiss Army knife. There's nothing there. The only thing that's even labeled is, there's the number of Qs, so this is a protein that doesn't have KD, because that's 20, but it could have been one with 46. For what we're talking about here, it doesn't really matter. And then there's this little blurb there that's called LBD, 3-letter abbreviations, for ligand binding domain. A ligand is something that binds to a protein. What binds to this protein is testosterone and dihydrotestosterone. So that's the ligand. And this is the site where testosterone or dihydrotestosterone would bind. And it's called, not the ligand binding domain, the LBD. Because this afternoon they're probably going to be saying LBD, 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 and now you know what it is. So let's bring in a and when it's like this protein does nothing. It floats around the cytoplasm, it lasts for a couple hours, gets destroyed, gets replaced. Does nothing. If you have testosterone, here we go. I feel so proud when I got that. It binds to the ligand binding domain because that's where it binds. And then you notice, I don't know if you noticed, but what would happen? It unfolded. One of the blades came out. And you see a new site you didn't see. So what's happening in the protein is it basically goes bump. And now there's new letters. NLS. Wow. That stands for nuclear localization site. And you're going to say, let's see, site, that's okay, nucleus. This is a site. It's a special sequence of amino acids in the protein that when exposed, wasn't exposed before, sends the androgen receptor to the nucleus. And there it goes. So whenever testosterone binds to the receptor, it opens up that site, and voila, it goes into the nucleus. Now we go to the nucleus, and we see now more blades came out. Now a whole bunch of blades. And I probably don't have them all. I'm only doing the ones I'm guessing you're going to hear later on today. Well, one of the things that it is, what testosterone and the androgen receptor do, is they go to the DNA and they turn on specific genes. Remember, in order to make a protein, you have to take the DNA and make a copy? The androgen receptor with testosterone starts that process at a specific gene. So what androgen receptors do when testosterone is on is cause you to make new proteins. That's what anabolic steroids do. You take them, they go into your muscles. Testosterone is the anabolic steroid. It makes your muscles make more muscle proteins, and you look like the governor of California. And so in order to do that, it has to bind to DNA. And so there is a special part of the androgen receptor that flips out. It wasn't open before, but it flips out when it comes in, called the DNA binding domain. BDD. BDD. DBD. I can't spell it. And it binds to DNA at a specific set of spots. Two other things that come out are a little bit more esoteric, at least for me. They're called AF2, AF1. There are other AFs, but these are the only one we're really going to talk about is AF2. These are special what are called activating domains. And they actually attract other proteins in the nucleus. And when those other proteins bind, they act as a co-activator with testosterone. They help testosterone cause the androgen receptor to turn on the genes. I don't know how to say it any other way. They're just kind of helpers. So in order for the androgen receptor to have its maximal effect, it can't do it just by itself with testosterone. It needs help. And these other helper proteins are called co-activators. And they bind to the AF2 and probably some bind to the AF1. In fact, the androgen receptor itself apparently can bind to the AF2. So it can either bind to itself, I guess like a dog with biting its tail, or another one can come in. I won't say what that might be like. So there's a whole mess of things. And every cell has different co-activators, so they activate different genes, and that's why it has different effects at different levels. And this is kind of a picture I stole. This is actually, I think it's a legal stealing because I got it from Wikipedia. And it kind of shows the whole process, but it's all static. Nothing moving. Here's testosterone or dihydrotestosterone. It binds to the androgen receptor. The androgen receptor goes into the cell. It binds to the DNA. And see all these other proteins. And notice what they are, abbreviations. CRB, SRC1, ARA70. All of them are those co-activators. They bind to those activator sites and they help the androgen receptor to activate DNA. And every cell has a different site. These are just three that are involved. One other thing I'm going to point out, because you're going to probably hear about it later on today, is that notice that on the androgen receptor there are a bunch of P's. Right there. Those are phosphate groups that were added to the protein when it went into the nucleus. And that's a common way to alter the way a protein works. It's called phosphorylation. And there's a whole lexicon. You can phosphorylate the protein. There's verbs, adjectives, that was done phosphorylate. Kind of pretty cool. I'm not going to talk much about it, but other than just remember that it does happen, and it can affect how the protein works. There are other things that can affect the androgen receptor that are added on like this in the same small molecules, and that's called acetylation. Again, you'll hear about it probably this afternoon, and they'll be able to tell you why they think it's important. And just realize this is a separate thing than these co-activators. These are actually glued on, small little activations. And they may or may not be involved in having Kennedy's disease, and that's, hopefully we'll hear about that tonight, or this afternoon. So, we have these 4 areas that flip out. Several researchers have demonstrated that if you remove or mutate the ligand binding domain, in other words, you go to the DNA of the androgen receptor, change the sequence of where the ligand binding domain is, that means testosterone doesn't recognize it and won't bind. It's kind of like a lock and key. If you change the lock, the key doesn't work. If you do that, then the androgen receptor never goes into the nucleus because it never binds to testosterone. And if you have an animal that has the KD gene, they don't ever get KD. So you have to have an active ligand binding domain and testosterone to have KD. That's why females don't have it. They don't have enough testosterone. Then, they also have changed, leave this one alone so it's okay, but then they change the nuclear localization site, the one that flips out. Get rid of that, it doesn't go in the nucleus, you don't get degeneration. So you have to have an active nuclear localization site in order to get Kennedy's disease. What if you change the DNA binding domain? You can guess it. If it doesn't bind the DNA, you don't get degeneration. So you have to be able to bind the DNA. Change the AF2 activation site, the one that interacts with all those other proteins, get rid of that, no degeneration. So it turns out that all these sites have to be in working order in order to have degeneration. If any of them don't work correctly, we don't get the degeneration. Now, what does that mean for us? And I don't know. It's not clear what that means. We'll hear some things again this afternoon about it, but remember, because you're going to hear about these. You're going to hear about these 4 sites and maybe a couple more. And so now you know that if you get rid of them, it doesn't work. So the protein has to work in order to cause the disease.
Do we know what ASCJ9 does?
I don't. But someone could tell. The JSC9, or you may know it. Heather?
Well, I think they answered in the slide. It sends it through the wood chipper.
Okay.
And how about the dutasteride? Which one did that affect?
Dutasteride was actually a little bit different. In this area, you see what you can have is testosterone, and in the cell it's converted to dihydrotestosterone. Dihydrotestosterone is quite a bit more potent. So if it binds, it's more likely to go through this process than if testosterone itself binds. Whatever, it's just not as potent. Dutasteride blocked that enzyme. So you wouldn't have DHT, you'd only have T.
But don't you need the androgen receptor to get strength?
Well, the idea was it would still work, it just won't work as well, and maybe that would reduce let me try it again. That would reduce the symptoms, but also it's also cell-specific. Some cells have it. I would assume it's a different form of the androgen receptor, so that it likes testosterone better.
No, well.
Or they don't have this enzyme, so it wouldn't be affected by testosterone. How's that one? Okay. I'll make up the answer until she nods. And so that means it wouldn't affect every cell. That was the good thing of it. It only affects certain cells. We were hoping it would be the right cells. Now, what do they think is happening? Well, again, it all depends on who you ask for. So I'm going to give the old standby. I'm going to bring up the proteasome. The proteasome is, as everyone who's seen this lecture before, is a wood chipper. The protein goes in one end and comes out as amino acids at the other. I can't help but think of Fargo for some reason. Now, it's thought That is, that when a androgen receptor that has the elongated, the poly-Q region, when it goes into the proteasome, it gets stuck. And it doesn't break it up. In fact, it makes a big fragment instead of only little amino acids, and then it spits it back out, or maybe even blocks it, so that it doesn't break down all of the protein completely. So there's always fragments left. The thought was, well, these fragments are now can't be destroyed because the proteasome can't destroy it because of this elongated poly-Q region, start to kind of join together, kind of stick to each other as they get more and more of them. And it's possible that those are actually toxic to the cell. In some way we don't know yet, but that's that accumulation. But as they stay around, they even clump, and eventually you get huge clumps of proteins that stick together. And that would include part of the androgen receptor and other proteins that just happen to get stuck. And the reason I think this might happen is because when you look in the cell, you see big clumps of proteins that have small parts of the androgen receptor and other proteins associated with it, as if it didn't get adequately digested. These are called nuclear inclusion and are characteristic of SPMA. So you see, these are basically remnants of proteins that didn't get digested well. That's how they return. If you want to see one up close, it doesn't look too much different than when it's far away, but it's these inclusions. So it's not clear that these are necessarily bad. This might be good because it might take the toxic parts away, but that's still isn't clear. The reason I'm telling you about this is because we're going to need to remember about conclusions in the future. Another reason why this is a very possible mechanism is because there's another way to destroy proteins. You can destroy proteins by putting them into something called the lysosome. And the lysosome is like a big vat of enzymes, and when something goes in there, it just gets destroyed. And the process by which it gets in the lysosome is called autophagy. Or autophagy. I'm not really sure which one's the correct one. But autophagy sounds better. If you activate autophagy in animal models that have KD, their symptoms, in many cases, go down, at least in the fruit flies. So that would agree with the idea that these extra pieces we couldn't get rid of, that we now find a way to get rid of them, voila. And I think that's what the ASCJ9 is involved in helping you get rid of. I think that's the theory. And maybe they don't know. I missed the talk. Now, so what we're going to do now is kind of more fun. This is, the people when they talk this afternoon are going to give you all the biochemistry, but in the end they're going to show you some sort of model which is going to try to mimic the idea of Kennedy's disease. One of the problems with Kennedy's disease is that no other animal that I know of has a CAG repeat disease. So if you want to study natural CAG repeat disease, we have to donate our brains. And they don't let us do that. It's just good. So what you have to do then is to try to make your own Kennedy's disease model in another animal. And you do this by taking the human gene with the elongated CAG and putting it into one of those animals. And amazingly, it works. And they get the disease. There are three major kind of classes that they may show today. One is cells. These are cells that live forever. You buy the cells. So it isn't like, they came from an animal maybe 20 years ago. And they have developed a model that you can actually take these cells, put in the androgen receptor gene, and it works. For example, this is a picture of certain cells that have androgen receptor gene, human androgen receptor gene. The cells are from a rat. And they make the protein. We know that because any place you see green, that's human androgen receptor. And when they add DHT, guess where the receptor goes? It goes to the nucleus. How do we know that? Because each of those little circles is the nucleus. So now we see that even though these cells don't naturally have a human androgen receptor, it works in a similar manner to the human androgen receptor, that when you put in a hormone, it goes into the nucleus. What if you have one with an elongated repeat? Well, very similar except for one interesting thing. Many of the cells have these very high intensity dots. Those are the inclusions. Remember the clumps. In other words, they're having trouble getting rid of the protein just like we think our cells have trouble getting rid of the protein. And so now we have something to measure. We can look and say, if we have something that has a long polycube, if we add something, can we get rid of the dots? And that might suggest that we're helping get rid of the protein and it might relieve the disease. There's a lot of ifs in there. Cells are the reason cells are used because they're cheap. Research is very expensive. This is a very cheap way of doing it, and it's a good first screen. But just because it works here doesn't mean that it works inside an organism. So you still have to put the gene into an organism. It's a little bit more complicated. One of the organisms that's used is amazingly fruit flies. To me, that was always amazing because fruit flies don't have testosterone. So if you give them an androgen receptor, that's the only one they have. And it can be done. Here's an experiment. This is taken from one of Paul Taylor's papers. And we're going to do the one on the left. The nice thing about Drosophila is that when you put in a foreign gene, the genetics is so well known, you can make that gene make the protein wherever you want it. So even though, in other words, when you have a gene for the androgen receptor, that doesn't mean the cell's going to make it. It has to have the right other proteins to turn on that gene. In fruit flies, you can make it, only make the receptor in a specific tissue. And in this case, it's in the eyes. And the reason they do the eyes is because it's really easy to see. Because fruit flies don't have it. So this is one that has the androgen receptor, which is a foreign protein to the fly, expressed in the fly. It has 52 Qs. That's wrong. It has DHT. Sounds like you should have Kennedy's disease. And when you look at the eye, you can see it's all kind of messed up. You'll see it going on the other one. This is what an eye should look like. And you can see that this eye looks messed up. If you cut it open, you see that it's degenerated. So the human androgen receptor in the fly causes degeneration just like it causes in humans, except, and only if you have the elongated polycube. This was an experiment done by Paul Taylor in which they altered, this is again the AR52, so that's the same 52. But now they made a mutation, and I believe this is in the DNA binding domain. So they changed the DNA binding domain so it won't bind DNA. Other than that, these two flies are identical. This one has degeneration, this one doesn't. So the conclusion is you need the DNA binding in order to get degeneration. And so you'll see a lot of this, because a lot of the stuff I talked about earlier about the different sites, some of that was done in fruit flies. And you'll be looking at a lot of eyes. So get used. Good, bad. Good, bad. Just like in this one, you want to see this. If you see these guys, that's bad. And then, of course, there's everybody's favorite organism, that's the mouse. So we have mice in which you put genes, human genes in them, they get Kennedy's disease. How do you tell that they have Kennedy's disease? Well, there's a lot of little tests. And this is the kind of cute part. For example, if you hold the mouse, and apparently yesterday you saw this already. You hold the mouse normally, a normal mouse, his legs go like this. And then he climbs up and bites you. This one goes like this. So it's a real easy test to tell if they have some sort of muscle problem. You can also do a cute thing, which is, except it's not as easy as it looks to me. What you do is you take the front paws and make them like red and the back paws blue in ink. Then you have them run on a piece of paper. And a normal mouse would actually, you can see the front and back paws are together. When they are. If they have Kennedy's disease, their feet are all over the place. So it's another way of telling if they've had muscle problems. There's something called rearing. Mice apparently will run around and look up and go back down and look up. And you can actually have little machines that measure how many times they rear. Well, if they're a normal mouse, they rear quite a lot. If you are a KD mouse, you don't do it as often because your muscles aren't as big. Then, of course, many of you remember the rotorod. The rotorod is a rod that turns. Initially slowly, and then over time it goes faster and faster and faster and faster and faster. And what you do is you time how long the mouse can stay on. The more normal it is, the longer it can stay on, the more the muscle degeneration, the less it's going to stay on. So if you do an experiment, the blue dots represent a normal mouse. After 7 months of training, they can stay on between 100 to 150 seconds, about 2 minutes. If you have a KD mouse, they steadily go down to where they're under 50 seconds. So it's another way of measuring whether or not they're improving or not improving when you give them a drug. So it's a very nice model. And I'm sure those who are in the trial, there are similar sorts of things in humans. How do you tell if a human is getting better? So we have the rotor rod for humans, and we have the hang test. What you don't see is Angela over here saying, "Hold on, you'll be sorry if you don't." I want to go over, this is the 10th anniversary of the KDA. We started in 2003 is when we gave our first research grant. And I want to kind of go over all those research grants, kind of like, well, look what we've done. And, you know, when you look over it, it's really impressive what those people have done with, you know, it isn't just our money, but our money certainly helped get them started. And so the first grant we ever gave was to Dr. Taylor, and this was to develop his Drosophila mutant. So all those Drosophila mutants you're going to see this afternoon, the KDA helped make them, because we gave them some money to get started. Well, Andy Lieberman was a little upset, I guess, so he decided he'd make his own model. So he was going to make a mouse model. So our second grant was for a mouse model. Most mice models up to that point, mice have testosterone. So they have their own testosterone receptor. And what most of the mouse models up to then were is that they would then add human receptors. So now they had 2 receptors, the normal mouse and the human. Well, what Andy did is he basically cut out the mouse one and replaced it with the human. So they only had 1 receptor. And this is called a not-in mouse. And the reason that it's good is that then you don't have another problem. Someone's always going to say, if you have 2 different forms of the receptor, well, it's not just the CAG repeat, something else is going on. So this is a model in which you're able to limit more like what it's in the humans. And so this is our second grant in 2004. 2005, another grant to Paul, and this is really to finish the beginning of the, fly model. In 2006, this is Dr. Chang. He's the one who did the JSCA9. And we helped with a grant in 2006 to get him started. And after that, he was able to get, I guess there's over $1 million grant now from NIH to treat it. And we had him at the ground floor. And that's pretty good. Une Pandi, who's in the back, you don't have to wave, but there he is. Who worked with, at this point, he was a postdoc in Paul Taylor's lab. In 2006, he also got a grant, and he was working with something called HDAC6. You don't have to worry about HDAC6, but what it did is it increases that autophagy. Remember, that's that alternate destruction pathway. And they were able to demonstrate that if you increase that autophagy, it rescued the cells. In 2007, Dr. Maria Frunoto, who's also in the back, but she wouldn't give me a picture of her, so the best I could do is the best I could do is show her that her work was published in Neuron, and not only that, it was on the cover. And if you look at the bottom, it says SBMA disarmed AR by IGF-1. And this was a study that was done to demonstrate that a protein called IGF-1 helps relieve the symptoms in mice. And it was good enough that it got on the cover. In 2007, well, Une's back. And it's again to continue his work on the front lines. And by the way, he now is at LSU in his own lab doing KD research, so we're spreading. And then we have, in 2008, Heather Monti. This is when she was in Paris. She was doing, remember I told you that when the androgen receptor goes into the nucleus, sometimes things get added. And one of the things that gets added is something called an acetylation reaction. And this is she was studying acetylation reactions. And I think we're gonna hear about them. We're gonna hear about them. And also I want to show you that sometimes when you do that transgenic mouse, you get really unintended consequences. I'm not sure what that is. But it's not a KD mouse. In 2009, Dr. Pasakandemi Johnny, I've been practicing that, who is with Alice Body here in San Diego. I think he's also going to be, I'm hoping he's going to be here. I'm not sure.
I haven't heard that he's coming.
And he just got the grant last year, and his job was, remember, we were unsure if the defect was because of the muscles causing the nerves to die, or the nerves are causing the muscle to die. He's going to develop a mouse model where the muscles don't make any engine receptor. So that way, if the muscles still die, it must have been because of the nerve. So it's a very interesting question, and hopefully maybe Alex will be able to see how we're doing on it. Maria's still not here, but this time she didn't get her work done, so she doesn't have a picture of the journal. Remember we said the other thing that happened when it goes in, you put on those phosphates, phosphorylation? She's studying enzymes that are involved in phosphorylation. And we will hear about some of them, right? Yes. And finally, Lenore, is another question that came up. Remember, is it true that when the AR goes in the proteasome, does it really get stuck? That's the theory. Lenore has divided the system, and she hopes we're going to answer that question and see if it really does get stuck. And so these are all research, and you notice that almost everything I talked about was somehow related to what these people had been doing. There's almost nothing that wasn't. And so it's really, I think, an amazing luck we've had with what we've funded, because the people have gotten really good research done. And we have over, these are the only ones I could find. I think there's more, at least 12 major research articles. And these aren't just in, Worm Runner's Digest. This is in Nature, in some of the major journals. And so we have been, I think, very good at spending your money on research that really seems to be making a difference. And, when I heard it was 10 years, all I could think of was a group back, most of us can remember, not in the back though, you're too young, about the group 10 years after, and their most famous song was "I'd Love to Change the World." And it's kind of, I wouldn't have given you the whole lyrics, but some of them really weren't too very appropriate. And the part, according to them, they didn't know what to do, but that's not true for us. We know that what we want to do is the next time someone comes up to you and says, we want to do something stupid. I want to run a marathon. I want to do a triathlon. I want to sell a cookbook. All for Kennedy's disease. What you're really funding is all that research that we did. And they really have done an outstanding job.